Biology Specimen 2016. Question 1. Figure 1.1 shows a light microscope. This is the microscope that we have. Okay. So let us first of all identify the parts of the microscope. So we have this is the eyepiece lens. And this is the revolving. And this is the objective lens. And this is the stage. And this is the condenser. And this is the diaphragm. And this is the mirror. And B, it is called coarse adjustment knob. And this is the fine adjustment knob. This is a clip. So A1, identify the parts labeled A and B. So we have identified A, it is a mirror. B, it is a coarse adjustment knob. Then part 2, using letter X label on figure 1.1, one part where magnification takes place. So magnification takes place here, which is the objective lens, even the eyepiece lens. So these two, that's where magnification takes place. So this is where magnification is taking place, or on the eyepiece lens. B, outline the correct procedure to follow in order to use the microscope to view the specimen. So to view a specimen, the first thing that we need to do, we need to put the specimen on stage and we clip it to avoid it sliding. Then from there now, we are going to view the specimen through the eyepiece lens as well as rotating the nose piece. And the third thing is we are going to adjust the coarse adjustment knob to bring the specimen into focus. And we use the fine adjustment knob to bring the specimen into sharp and clear focus. So that is the procedure that we need to follow. So put the specimen on stage, clip it, view the specimen on the eyepiece lens, and rotate the revolving nose piece. Adjust the coarse adjustment knob to bring the specimen into focus. Adjust the fine adjustment knob to bring the specimen into sharp focus. But see, describe how magnification is determined when using a microscope. So how we determine magnification is that we multiply the power of the eyepiece lens by the power of the objective lens which is used. So that is how we can determine magnification using the microscope. So you determine magnification by multiplying the power of the eyepiece lens by the power of the objective lens used, i.e. if the power of the eyepiece is 10x, you multiply with 40x, maybe the one which is used for objective lens. So that's how we can determine. So number 2.1 shows part of the alimentary canal and its associated parts. So this is the liver, we have the gallbladder, the pancreas, the duodenum, the esophagus, and we have the stomach. A1. Which of the labeled parts in figure 2.1 are bile salts produced? So bile salts are produced in the liver. So the liver is the one that produces the bile salt. Okay, the gallbladder, it just stores. It stores the bile. By the bile, it is produced by the liver. So the labeled structure, it is the part C. Part 2, explain the role of the bile salts in digestion. So the role of the bile salt in digestion, it is to emulsify the fats. To emulsify the fats. So emulsification, it is the breaking down of large fat droplets into smaller droplets for enzyme efficiency. So before the digestion of fats begins, the first thing, fats must be emulsified. So what emulsify the fats? It is the bile salt. So that is the role of the bile salt. B1. In which labeled structure in figure 2.1 are proteins first digested? So proteins are first digested in the stomach. So the stomach, it is the first part where proteins are first digested. So the answer is E. B2, explain how protein digestion takes place in the structure identified in B1 above. Protein digestion in the stomach, how does it go about? So the first thing is the walls of the stomach, they will secrete what we call the gastric juice. So gastric juice, it contains hydrochloric acid and it contains an enzyme which is inactive, pepsinogen. So hydrochloric acid, it is going to activate pepsinogen to pepsin. Then pepsin, which is an enzyme, it will convert proteins to peptides. So that is what is going to happen. So the juice that is secreted in the stomach, it is gastric juice. It's a one which contains hydrochloric acid and pepsinogen. So pepsinogen, it's an enzyme which is not active. So it will be activated to pepsin by hydrochloric acid. Okay, then pepsin, it will convert proteins to peptides. So part C, name two common elements of structure C. So structure C, which is the liver. So the liver is structure C. So the elements of the liver, number one, it is peptides. Number two, it is hepatomegaly. Number three, it is cirrhosis. So these are some of the diseases of the liver. But if they said effects of the ailments, would have said swelling of the liver, would have said abdominal pains, would have said death. But here we are just concentrating on the common ailments of the liver. Number 3. Figure 3.1 shows a section through the heart. A1. From the figure, identify the two blood vessels labeled G and I. So G, this is the vena cava. I, this is the aorta. You can also identify J, which is the pulmonary vein. Then H, it is the pulmonary artery. Then we have valve K, which is aortic semi-runner valve. Part 2. Which of the labeled blood vessels carry oxygenated blood back to the heart? So back to the heart, the one which carries oxygenated blood, it is pulmonary vein from the lungs. So that is the answer. So there it is J. Part 3, identify valve K and state its function. So valve K, it is aortic semi-runner valve. So aortic semi-runner valve. So the function of the valve, it is to ensure that blood flows in one direction or to prevent the backflow of blood. Part B, describe the movement of blood from the time it enters the heart at J until it exits at I. So J, this is pulmonary vein. So blood will enter through J, which is oxygenated blood. So oxygenated blood from the lungs will enter the heart through J, which is the pulmonary vein. Then it will come left atrium. From the left atrium to the left ventricle. From the left ventricle, then it will go to all parts of the body through the aorta. So that is the movement of blood. So blood move into left atrium through J, then moves to the left ventricle. From the left ventricle, blood is pumped to all parts of the body through I. Part C, some poor diets can increase the risk of a heart attack. Suggest 2 other factors apart from that can increase the risk of a heart attack. So number 1, it is smoking. Number 2, emotional stress. Then we have lack of exercise. So there we cannot include excessive intake of fats because they've already mentioned it there. Number 4, figure 4.1 shows a root system of a leguminous plant. So this is the plant that we have. A1, identify structure L in figure 4.1 above. So this is what we call root nodules. So L, it is called root nodules. So we can give an example of leguminous plants such as beans, such as groundnuts plants. So on the roots, there are these which we call the root nodules. Okay, so part 2, state the microorganism found in structure L. So the microorganism that is found there, it is the bacteria called Rhizobium bacteria. So part 3, explain the role of the microorganism named in 2 above, a nitrogen cycle. So what is the role of Rhizobium bacteria in nitrogen cycle? Its role is in nitrogen fixation. So this bacteria, it is going to trap nitrogen from the air. Then that nitrogen to be converted to ammonia. Then ammonia, it will be converted to ammonium compounds for plant use. So that is the role of rhizobium bacteria. So in nitrogen fixation, so the bacteria traps nitrogen from the atmosphere into the soil. It is then converted to nitrogen compounds. Part B, explain the role of this plant in figure 4.1 plays in the carbon cycle. So in the carbon cycle, number one, the leguminous plant, it takes in carbon dioxide from the atmosphere for photosynthesis. Okay. Then the second one is that it is through respiration. So whereby it gives out carbon dioxide to the atmosphere by the process called respiration. So that is its role in the carbon cycle. Number two, explain the role of this plant in figure 4.1 plays in the water cycle. So in the water cycle, it plays a role of transpiration. So where the plant, it is going to absorb water from the soil and it will lose water to the atmosphere through water vapor. Number five, blood group inheritance in humans is controlled by three areas, A, B, and C. Using appropriate genetic symbols, draw a genetic diagram to explain the possible blood groups of children whose parents are both heterozygous. For their blood groups, the father being blood group A and the mother blood group B. So now what does it mean when we say heterozygous? So heterozygous, this is when we have a genotype of different alleles. So different alleles of blood group A. So different alleles of blood group A. So blood group A, it is AO. That is heterozygous. If it was homozygous, blood group A, it is AA. Okay? So what is the genotype of heterozygous of blood group B? So blood group B heterozygous, it is B O, but for homozygous, it is BB, that is blood group B. So now, how do we make the genetic diagram? So you start with the phenotype, which is the outward expression. So phenotype, we have the father who is blood group A. Then we have mother So that is our phenotype. We have father of blood group A. We have mother of blood group B. Now we have genotype. So genotype, remember heterozygous of blood group A, it is AO. So this is blood group A, heterozygous. Then mother blood group B, heterozygous. So that's what we have. So if it was homozygous, it would have been BB. A. That's what it means. Then we have gametes. So we cross. So this it goes with that. So what we have, it is A, B. Now this goes with this. So what we have, it is A, O. And now O goes with B there. So what we have, it is B, O. Again, O goes with O. So what we have, it is OO. So that's what we have. So these are offsprings. So these are offspring genotype. Now we have offspring phenotype. So AB, that is blood group AB. AO, that is blood group A. BO, that is blood group B. Then we have OO, that is blood group O. This is what we have, and you'll be awarded 5 marks. So for the offsprings, we have blood group AB, we have blood group AO, which is A, blood group BO, which is B, blood group OO, which is O. So those are the offsprings phenotype. Part B, using a named example, explain continuous variation. So continuous variation, this is a variation where a characteristic has many intermediate forms between two extremes. That is what we call continuous variation. So examples of a continuous variation, we can talk of complexion, body size, the height. These things, they do change due maybe to environmental factors, or it can be controlled by many genes. So that is what we call continuous variation. So continuous variation, it is affected by environmental factors such as diet, such as the temperature, humidity, so on and so forth. Also by many genes. So those are the factors that affects continuous variation. So examples, then we have now discontinuous variation. So discontinuous variation, it's a variation in which a characteristic has clear-cut different groups with no intermediate forms. So in other words, these are things that cannot change. For example, the sex of a person cannot change. The blood group of a person cannot change. We can talk of the color of the eyes, tongue rolling, those things, they cannot be affected maybe by environment and the genes. So that's the way it is. So those are examples of discontinuous variation. So discontinuous variation, it is not affected by the environmental factors. Then it is controlled by one or few genes. Section B, 6A, describe the following. Number 1, common causes of blindness. But one cause of blindness is injury to the eyes. Injury to the eye. So injury to the eye, it can cause blindness. Then number two cause of blindness, it is the deficiency of vitamin A in the body. So this, it can cause night blindness. So deficiency of vitamin A in the body, it can cause blindness. Apart from that, we have diabetes. So diabetes retinopathy. So it can cause blindness. Apart from that, we have what we call bacterial infection of the eye, which is called the trachoma. So that is called trachoma. Okay. So now part 2, describe the following common methods of preventing blindness. So how can we prevent injury to the eye? It is by wearing, those who are working, by wearing the eyewear, which is the protective equipment. By also eating foods that are rich in vitamin A, also by controlling diabetes and the high blood pressure. Okay, so by also keeping good hygiene to prevent infection to the eye. But B, describe the effects of abuse of a named drug on the nervous system. So the named drug we can name alcohol. So what about alcohol? So alcohol, the effect of alcohol on the nervous system, number one, it causes the loss of coordination. Number two, slowing down transmission of nerve impulses. So this, it slows down the reactions to stimulus. Not forgetting loss of memory. So alcohol, it also damages the brain cells and also slurred speech. These are some of the effects to the nervous system. So if it was just generally, its effect would have added, it brings about poverty. 7a, describe the processes of fertilization and implantation in humans. So fertilization, it is the fusion of the male gamete and the female gamete to form what we call a zygote. Then this fertilization, it occurs in the oviduct, which is called the fallopian tube. That is the fertilization. So that is called fertilization. The second one, it is implantation. So implantation, it occurs after fertilization, which is 6 to 12 days of fertilization. Then we have implantation. So implantation, it is a process by which a zygote It moves from the fallopian tube and it is attached to the walls of the uterus. So that is what we call implantation. So implantation, it occurs after fertilization, 6 to 12 days of fertilization. So that is implantation. Part B, describe ways of maintaining a health pregnancy and the safe childbirth. So number one, how can we maintain health pregnancy? So we can maintain health pregnancy by eating a balanced diet. That's number one. Number two, by managing stress. Okay. Number three, by avoiding taking harmful substances such as alcohol and cigarette. So those are some of the ways we can um, use to maintain a health pregnancy. Also by prenatal care. So that's how we can maintain health pregnancy. Prenatal care, managing stress, taking balanced diet, prenatal care, avoiding taking harmful substances such as alcohol and the cigarette. Now, what about maintaining safe childbirth? So we can maintain safe childbirth, number one, by attending childbirth education classes. Okay. Also, postnatal care. Not forgetting delivering in a qualified health care system. Number eight. A. What role is played by the following structures of the skin in controlling the temperature of the body during overcooling? Number one, erector muscle. Number two, blood vessels. Part B, explain the metabolic functions of the liver. The erector muscle. So number one, the erector muscle contracts. That is during overcooling. It contracts and pulls the air upright. Then the air it will begin to trap the cold air on the surface of the skin, thereby reducing the loss of heat. So number two, blood vessels. So blood vessels during overcooling, the blood vessels they constrict, meaning they become narrow, preventing the flow of blood near the skin, thereby preventing the loss of blood through convection and radiation. Part B, explain the metabolic functions of the liver. So number one, the liver, it regulates the levels of glucose in blood. That's number one. Number two, the liver produces the bile, which is used for digestion. Number three, the liver detoxicates the poisonous substances such as alcohol and drugs. Then number four, the liver, it produces it, which helps in thermoregulation. Number five, the liver also diminate amino acids. So these are the functions of the liver. So dimination of amino acids, regulates the level of glucose in blood, generates it through metabolic reactions, detoxification of harmful substances such as alcohol and drugs, production of bile used for digestion of fats. 9a. Describe the following deficiency diseases in plants. Number one, chlorosis. Number two, leaf freaking. So chlorosis, it is the yellowing of plant leaves due to the lack of chlorophyll. So chlorosis comes about due to deficiency of nitrogen and magnesium. So that's how it comes about. So deficiency of nitrogen and magnesium causes chlorosis. Then leaf freaking, this is a condition where there are some decolored spots on the leaf due to lack of some nutritional components in the plant and other factors. Part B, discuss the functions of the nutrients phosphorus, nitrogen, and magnesium in plant growth. So the function of of phosphorus, it is for flowering in plants, for fruiting, not forgetting for root development. So that is the function of phosphorus. Then we have nitrogen. So the function of nitrogen, it is for general growth of plants, okay, which are leaves and stem. Then the other one, it is chlorophyll production. Then we have magnesium. So magnesium, it is the main essential element for chlorophyll, not forgetting photosynthesis. So magnesium, it is also used for activation of enzymes. Number 10A, discuss the importance of the following responses exhibited by plants. Number 1, phototropism. Number 2, geotropism. Number 3, hydrotropism. So the importance of those. So phototropism, it is the response of the part of the plant by either growing towards or away from light. So the importance of phototropism, it is to expose the leaves to sunlight for photosynthesis. Geotropism, it is the response of the part of the plant by growing towards or away gravity. That is geotropism. So the importance of geotropism, it causes roots to grow downwards in search of nutrients, water, and anchorage of roots to the ground. So geotropism, it causes roots to grow on the ground to search for water, which is used for photosynthesis, nutrients, and anchorage of the plant by the roots. Then we have number three, hydrotropism. So hydrotropism, it is the response of part of a plant by growing towards or away water. So roots, they grow towards water to absorb water for photosynthesis, for plant survival, which is for cooling and transportation. Part B, state and explain task responses exhibited by invertebrates. So state and explain task responses exhibited by invertebrates. So number one, we can talk about phototask. So phototask or phototaxism. So phototaxism or phototask, it is a response of an invertebrate animal by either moving towards or away a stimulus which is called the light. So example that we can give is cockroaches. They respond negatively to light. So that is phototask. Then the second one that we can give, it is chemotask. So chemotask, it is the response of an invertebrate by either moving towards or away chemicals. So moving towards chemicals, it is called positive chemotask. Away from chemicals, it is negative chemotask. Then we have also what we call thermotask. So thermotaxism, it is a response of an invertebrate by either moving towards or away temperature. So moving towards the temperature, it is called positive thermotaxism. Away from temperature, it is called the negative thermotaxism.